Hello, and welcome to our webinar, Fueling Discoveries and Introduction to the ALS Global Research Initiative, also known as AGRI. So on today's call, we'll take you behind the scenes of AGRI to learn exactly what it is and how your participation makes a real-time impact on ALS research. So we will be sharing a recording of the webinar afterwards, along with a link to the most frequently asked questions. Thank you to all of you who submitted questions in advance. And if you have any questions that come up during the webinar, feel free to use the Q&A module in the Zoom chat. Additionally, I just want to give a reminder that personal medical questions should be referred to your physician. And with that, I want to say that today I'm joined by Laura Dugom and Dr. Björn Oskarsson, and I'm going to let each of them introduce themselves and get us going. Laura, go ahead.
Thanks, Stephanie. I'm Laura. I'm the clinical research scientist at the foundation. Before I came to Target ALS, I worked in the pharmaceutical industry, primarily on phase 1 through 3 clinical trials and post-market approvals in rare diseases. And I'm really excited to be with Target the last 2 years and introduce the ALS Global Research Initiative to everyone and share a little bit more about our natural history study, the impact it's having on ALS research, and how your involvement can make a difference. Bjorn?
I'm Björn Oskarsson. I'm a neurologist at the Mayo Clinic in Florida. I direct the ALS Center of Excellence here. I have an interest in ALS care and ALS clinical trials and biomarker collection and trying to understand what ALS actually is. So I'm very excited about getting to participate in the Target ALS program and have had the opportunity to do so for a couple of years. So very excited about getting to participate in today's webinar.
Thank you both. Okay, I'm going to get the presentation going here. Can you see this?
Yep.
All right.
All right.
Laura, will you take us away?
Sure. We can go ahead and go on to the next screen. So before I talk about our research studies, I really want to share a little bit about who we are as an organization. Target ALS is the largest funding organization in the world for ALS research, and our vision is that everyone lives. So we're focused on breaking down barriers in ALS, and we work around the clock to carry out this vision. And we do this in 3 ways. We have the researchers and grants we fund, the work we enable through our resources that we make available to the scientific community, and the clinical research we conduct. And today we're going to dive more so into the research we conduct, which is AGRI and our global natural history study. But I want to pause and say that this urgency we all feel today on this call and while we're here is to do more to understand ALS and design better clinical trials. And that's exactly why our global natural history study exists. And this research can move faster with your help. So I've mentioned AGRI a couple of times now, and AGRI is our ALS Global Research Initiative, and it's the most comprehensive and diverse ALS initiative. It's made up of 2 studies. We have our longitudinal global natural history study, and we also have our community-based pop-up clinics. Our goal is to apply long-read sequencing and longitudinal data collection to over 7,000 individuals globally. And what that means is Target ALS, we recognize that the disease doesn't know borders, so neither should the research. And so while these are 2 separate studies, they're both designed to be inclusive and bring research directly into communities so people with ALS and their families have real opportunities to contribute. So instead of working in silos, Target ALS is building a global network of data and samples to identify risk factors, better understand the disease progression, and help develop biomarkers. And we're working together on a global scale to answer all of these questions. I want to highlight actually our most recent pop-up event that we did in collaboration with ALS Arizona this week, where we enrolled 25 participants in under 4 hours this Monday. And I think that just shows the commitment of the ALS community to show up and drive research forward. All of these samples that we collected during our event on Monday are actually already at our partnering lab for sequencing. So that's the real-time impact of Target ALS. We don't let your samples sit on a shelf, and we're not just saying, but we're demonstrating how we are breaking down those barriers to ALS research. And I know that you might be thinking like, well, that's great, but we need to move faster. And we totally agree. So the reality is the speed and scale of this effort is possible because of people like you. And everyone on this call is in the fight against ALS. So if you choose to be a part of this research, every sample donated is moving us forward. And there's actually a QR code on the screen here that takes you to our AGRI page if you want to learn more about our research studies. And with that, I'm going to hand it over to Dr. Oskarsson to talk a little bit more about what a natural history study is.
So this study is a product where we're learning about people with ALS, where kind of how the disease starts, how things develop over time. And we check in periodically, both with clinical measures and these biosamples. These biosamples, again, of blood and spinal fluid, urine, where we can kind of check in on the disease at different times. Our current definition of ALS is really one based on where in the nervous system the problems are, and not one that we can really link to a particular protein or other biological abnormality. To get to that point, which is probably where we are going to be able to find really effective treatments, we need to do this groundwork. Again, shouldn't be, even if we want to move fast, we need to get this groundwork down first. Biomarkers are kind of what we're hunting for here. And for anybody with ALS, for many people at least, the diagnostic journey at first was long and sometimes confusing, where you might have seen several different specialists before someone finally landed with the diagnosis. That again reflects our current understanding of the disease, or defining it, saying that it's there based on certain findings on the clinical exam where we see problems in the nervous system. But if we had a blood test that could tell us that ALS is the problem, that would help us get to a diagnosis much quicker. It could also help us make the diagnosis correctly. Again, sometimes getting a ALS diagnosis of somebody in error is a big problem for that person, and certainly often the diagnostic delay, which is much more common, is also leading to problems, unavoidable problems for that person. Another question that many people with ALS ask is, what's going to happen to me next? How long will I live with this disease? How will things play out? And we can make guesses. I think many expert clinicians have ideas about this. There's some ways we can model prognostication and kind of predict the future, but we're not very good at it yet. And this is something that we need to improve, both to directly answer people's questions now, but also it can help us for response markers. Again, these therapeutic biomarkers, where if we're testing a drug in ALS, now right now it's really hard for us to see if the drug has a meaningful benefit. The measures we have today for trying to see if something works or not are pretty blunt. And we need to select people for clinical trials that are early in their disease and where things are changing rapidly for us to be able to kind of get meaningful data out of it. And that is very frustrating for most people with ALS who cannot participate in clinical trials. So your participation wouldn't help us answer the question. And it's frustrating for the researchers who, again, we have to turn people away who want to participate, who we want in. The drug companies, this means that trials take much longer to enroll and are much slower to run, which makes them much more costly. So this is something where if we can have good blood markers that can tell us if a drug is working, that would help so many things. Now, we're not at scratch today. We do have some stuff going on, and there has been improvements in the last few years, but we're still not where we want to be. We want to be able to do this quicker. Over to you, Laura.
Yeah. So, and to elaborate a little bit on that, I want to share more about the global impact of our natural history study so far. So in the last year, we've more than doubled the number of sites that we have activated, and had our first international sites activated last year in South America, and then this year in South Korea and Israel. And so we have 14 active sites globally, and we're working to bring up newer sites in India, Malaysia, the Philippines, and Indonesia. But we're already learning and seeing the differences globally. There's differences in disease progression. The mutations that are more common around the world are different in different parts of the world. And we're already learning these things through our study. So I mentioned that AGRI is the most diverse initiative. We currently have 260 participants enrolled. Our goal is 1,000. But we have 34% of those 260 participants that come from diverse backgrounds. And to put it in perspective, most ALS studies only have about 5% of its participants coming from diverse backgrounds. I also mentioned that it's the most comprehensive initiative. And so what we're doing is looking at um, different visits and collecting samples longitudinally. And by doing that, we're making these samples available to researchers and the data and available to researchers in real time. And so far, we've collected over 26,000 biofluid samples that are all available in our repository in Arizona and have distributed over 3,000 of those samples. And so all of that data that's collected in parallel with those samples and entered by our amazing coordinators is shared in real time to the research community through our Target ALS data engine, which is controlled end to end by Target ALS. We've seen the data come in, we're putting the data out, and it's a very powerful resource that we have to enable research. And so we can go on and talk a little bit about, okay, that's the natural history study at a high level. What if I want to participate? What impact can I have? And what does that experience look like for me? So if you choose to participate as a person living with ALS, you're going to be asked to come to clinic for up to 5 study visits. Each visit is about 4 months apart. And you're going to be asked questions about your health, asked to donate biofluid samples. And a lot of these questions are actually the same types of questions and assessments that you would have done at a regular routine ALS clinic visit. So that's things like your ALS FRS or your vital capacity. You're going to also be asked to complete a one-time environmental questionnaire. And the purpose of this is it's going to help researchers better understand what are the possible factors outside of your genetics that might be impacting your disease. And it's asking a lot of important questions that might help us better understand a lot of the things that Dr. Oskarsson was pointing to. So, like disease progression over time. And then there's also optional at-home speech and motor function assessments that you can opt in to take. And you're going to do those from home about twice a month. But to help overcome these barriers to research, Target ALS covers the cost for you and your caregiver to come to clinic for every one of these visits. So that includes things like your hotel, your transportation, and food. So we're really trying to make it accessible for everyone that wants to be a part of research. And I'm going to also turn it over to Dr. Oskarsson to talk a little bit more about the biofluids we're collecting and why they're important, and maybe some misconceptions that people might have about spinal taps.
Yeah, we in the end want products that are easy to use, and spinal taps and spinal fluid is a little bit more complicated. But from where we're standing today, it is what we need to understand what's going on. Again, ALS is a disorder of the nerve cells in the brain and in the spinal cord. They're separated from the blood by the blood-brain barrier. So we can't really see what's going on in the brain and nerve cells by looking at the blood. Eventually, the spinal fluid is filtered into the blood, and it's being cleaned there by the livers and the kidney, and we might catch something in the urine and so on. But it's much harder, and we're looking at the problem through a filter. So we need to be able to understand and see what's going on in the sick nerve cells, which when they're sick, spill a lot of things, proteins, into the spinal fluid. We need to look at the spinal fluid. Spinal fluid collections are more complex than blood draws, but they're not tremendously much more complex. The way that the humans are made, we have a spinal cord that ends around your breastbone, and the rest of your spinal column is a fluid-filled space. So from kind of mid-chest down, it's just empty space. There's lots of fluid in there, and we can insert a needle between the vertebrae and get there. The backbone there is a very sturdy structure, and there's not really anything else there. So it's relatively easy to get a needle in there. The way that we do this carries a slim risk of headaches. The current needles that we use nowadays are much, much thinner than the needles we had 5 years or 10 years ago. There used to be a risk of a headache after a spinal tap of maybe up to 10%, and now we're down to about 1 in 100. These needles that we use, they're very thin. The actual hole inside the needle is essentially hair thin. So it does take a while for the fluid to run out of the needle. So it's a little bit boring there to get the fluid out, but it does really cut down on the risk of developing a headache. So those risks have gone down over the years since I got into this field. And the reason why people can get a headache afterwards is that, again, this fluid normally floats your brain. So your brain normally kind of floats in this fluid. And if you have too little fluid and you stand up, then the fluid will run down into this and not quite float the brain. The headache would go away if you lie down, but it would come back if you sit up. The amount of fluid we take in this study, you make that up in about 45 minutes or so. So it's not a loss of spinal fluid that takes a long time to recover from. And that is really the one risk that we see with the spinal tap. There's not any risk of paralysis or infections. As a physician practicing in the US, yes, we will put those on the potential risks with any procedure. But the one thing that we worry a little bit about with this procedure is headaches. And with modern techniques, the risk of that is really low. These procedures are generally done by neurologists, normally your own neurologist. Here at Mayo Clinic, we have the nurses do the taps. Again, it's really not that big a deal, but and when you sign up for this program, I mean, we're asking you for a commitment to do several of them. But if in case you change your mind, if it wasn't a good experience, you have it's absolutely okay to again, step back and not continue, but it's very rare. People who give once tend to come back. The first time is the scariest.
Thanks for sharing that. And I think a good thing to highlight too for any of the biofluids, if you decide, that time, you don't feel like doing the CSF that day, or maybe you want to come back another day within a month of that visit, you're more than welcome to do that. I mean, at any point in the study, you can choose to not do an assessment. So I think that's a really good point. We can go on to talk about healthy controls and what that experience looks like for participants. So the way we define a healthy control in our study is someone who does not have a neurological disease. And most of the time, our healthy controls are caregivers, families or friends of loved ones. And at the time of consent, we just ask that if they know if they have a genetic mutation or not. There are other studies out there that are looking at people with mutations that are asymptomatic, but our study is looking at people who, at the time of consent, don't have a known ALS mutation. And so a lot of the assessments you'll see here, they're the same as for the ALS participants, just less frequent. So you only have 2 visits as a healthy control, a baseline and then a visit 2, and it's about 12 months apart. The travel reimbursement policy is the same. So any way we can help get you to overcome that travel barrier to research, whether it's, an Uber, or plane ride, or hotel costs, we do that for our study. And we're aiming to enroll about 200 healthy participants. And we're actually about halfway through, which is really exciting. And that just shows that you don't need to have ALS to impact research and drive it forward. And controls are really important for lots of reasons. So ALS is a heterogeneous disease. And what that means is that people progress differently. Their symptoms of onset are different. And so it's hard to understand what ALS looks like in everyone. And a healthy control becomes a sounding board for scientists because it allows us to better understand what are the potential biological changes that are at play that we could link to ALS. And that gets us closer to a lot of the things that Dr. Oskarsson was talking about, like a blood-based biomarker. And we can move on. So I wanted to share, and this is honestly the best part of research, I think. I'm not going to read all of these quotes, but I want to highlight one. So, Margaret, who was one of our participants who was living with ALS, she said, "You're either the conduit of progress or an obstacle." And I think that's really important for people living with ALS and for the scientific community as well. And honestly, we're just so grateful for every person who decides to be a part of AGRI or our natural history study. And we want to make sure that action always turns into impact. And so that's why we built AGRI differently than a lot of the other studies that are currently ongoing. And I'll turn it over to Dr. Oskarsson to talk about some key differentiators of this initiative.
Well, yes. The so this study does collect a large number of fluids over a long period of time. The spinal fluid, as we touched on here, is kind of the key. It is the fluid that is generally the hardest to find for researchers trying to look into ALS, to look for ways to find a cure. And having this available, easily available, coupled with a really well described story of these people who have given their samples is very valuable. Again, without these samples, it's not possible to do experiments. The diversity aspect here, again, we've already heard about. ALS is highly variable, and we get additional insights by getting this broader look at the different facets of ALS. There are other projects out there that collect samples for researchers. Most of these projects are, say, coupled to Mayo, so we have our own projects that have been running. It's really hard to get those samples out to researchers at, say, Emory or Mass General or Hopkins when we want to collaborate with them. We do when we get it out there, it works, but it takes time. Here with Target, that's already taken care of up front. And so the samples go out right away without a lot of legal arguments, and it is really it's much faster. And then the quality of the samples, again, it's really processed in a very systematic way where each one of the sites do things exactly the same. There's also this kind of, I don't know, metadata is maybe not the right word for it, but other coupled data points, like the neurofilament light data that's coupled with these samples and multi-omic data sets. So you would, when you want to do an experiment with these samples, you would be able to kind of see and learn a lot about these samples, not just the clinical aspect of the patients who gave them, but on other measures that you're probably going to be thinking about and then wanting to select patients based off on. So it is really makes this data set richer and more powerful than most others, which would lack many of these things. We unfortunately see that many of the clinical trials that we do in ALS don't bear fruit in the form of success and approved product. The samples given to a drug company may or may not be shared widely. Often the answer is not. And then those samples are just sort of put away and not utilized for ALS research. Here, anything you give will be used for ALS research. And that is very empowering. There are many people who want to take the fight to ALS, and this is a good way of doing it.
Yeah. And I also want to add that all of the samples that we distribute through our biofluid repository is shared with researchers with no strings attached. So anyone who receives our samples anywhere in the world, they don't owe us anything. We're all here to help move it forward.
Thank you both for that great presentation. So I'm going to stop the share now and we're going to get into some Q&A. All right, so first off, we did get a question about neurofilament light. So Dr. Oskarsson, I'm just going to ask you, what is NFL and how is it used as a biomarker for ALS?
The neurofilament is a very interesting, it's a group of proteins. They're scaffolding proteins. They're sort of building skeletal proteins almost of the nerve cells. So they're not it's a protein that's involved. These are proteins that are involved in any nerve issue, really. So it's not specific to ALS. It's not just ALS that shows up with high neurofilament light values, but at first we could capture neurofilament values in the spinal fluid. And so we learned there that it came up in ALS, and the faster the ALS went, the higher the levels were. Now, with additional technique development, it turns out we can actually measure this pretty good, or even better, in blood. So we do both still, but it's really become one of these translational successes, what we've taken from spinal fluid and moved it into blood, which is much easier, and with some tweaking there, it actually works better than in spinal fluid. It, again, can help make a diagnosis, but again, it's not specific for ALS. There are lots of other diseases. Some diseases even drive it up higher than ALS, but ALS drives it up high. It does speak to the speed that the ALS is eating at the nerve cells. So it's a prognostic marker. And the drug for SOD1 ALS, they call SODI drug, that's really been a game changer for that portion of people with SOD1 ALS, was largely approved based on the neurofilament light levels. So it is starting to be used in clinical trials, and our regulators are starting to look at it with some confidence. There's still a little bit of uncertainty about the marker. There's still uncertainty about the marker. There's still things we need to figure out about what pitfalls there might be with using it and so on, but it is very much right now front and center, and it's being used across the board in most ALS projects.
Thank you. Yeah, so it seems like very promising and exciting, but of course we have to search for more of those biomarkers of those different types that you spoke about earlier. Next, we got a couple people asking a couple questions about an observational trial like this versus an interventional trial. So Laura, why should someone consider participating in an observational study?
That's a great question. So actually, a lot of the times, interventional studies may allow you to be on an observational study at the same time as an interventional one. So first, I would say to ask your clinician, could you be on both at the same time? And if so, then it's completely your choice if you want to. And so an interventional study is exactly that. There's some sort of treatment being administered, whether it's placebo or something that we're testing to see if it's the efficacy in whatever disease you're looking at. So with ALS, there are a couple of clinical trials where, there's ASOs, for example. And so that's an intervention. Whereas a natural history study, we're really looking at the disease progressing naturally. So if we do nothing and intervene with different medications or testing medications, how is that disease changing over time? And so that's what our natural history study is trying to answer.
Thanks, Laura. Next, we've had a lot of questions about the genetic causes of ALS. We know some of them, like C9orf or SOD1. Are we learning more about genetic mutations that will help us better understand sporadic ALS? Dr. Oskarsson, would you like to answer this question?
Sure. I mean, I think our best current models suggest that there is some genetic contribution, to a much larger portion of ALS than what we can test out right now with single gene contributions. The links we see today are where there's a single gene that's changed, and people with that change have a much higher risk of getting ALS. But one can imagine that there are problems, maybe double trouble, where you have two different genes that are together interacting and leading to disease or increasing the risk for disease. So we're hunting for those. And this, I think Laura mentioned long read sequencing as something that was rolling out here. We can look at the genome in many different ways, and we're getting better and better at looking at it and understanding it. And this is the latest and greatest kind of way of looking at it. Hasn't really been done at scale. So that's something that's being done here. And that probably will uncover some new things. So it is still something where we are looking, and we're expecting to find more. There's still going to be a large portion of ALS based on our best models today that don't have clear genetic causes. where there's more environmental factors that are at play, and trying to track that down is equally important. And even if you have a gene that puts you at risk, an increased risk of coming down with ALS, there's still lots of people who have one of these genes and never get ALS. So there's probably these other factors are probably important for people who have a risk gene also. So it's probably not for most people one single thing that drives the disease, but several factors.
Thank you. Laura, anything to add on that?
No, I echo everything Dr. Oskarsson said.
Thanks. And those environmental factors, I think we always hear about things like veterans are more likely to get ALS, or sometimes it's like long distance runners or people who exercise a lot. There's been some conversations about like where you live, if certain bodies of water, things like that. How is this study helping identify those environmental risks, or like confirm if those things that we're hearing are true? Laura, would you like to start?
Sure. So we don't know, right? So when we developed our environmental questionnaire, we put a bunch of different questions that start at a high level. So over time, as we collect more data, we can kind of narrow it down and better correlate things if there is anything to look at. So we ask a lot of those questions about, have you served in the military, education, past injuries you may have had where you've ended up in a hospital or had maybe a brain injury in your childhood. So we're trying to get a holistic snapshot of the whole exposome, which is things outside of your genetics, to better understand how we can target these questions better, to know how it correlates to ALS.
Thanks, Laura. Dr. Oskarsson, anything to add there?
No, I think that, and again, this complements other efforts. I mean, when we're trying to look at this, the more data sources we have, and again, this is such a fantastic resource since you also have the biosamples. It's not just the environmental questionnaires, but it's coupled with the biosamples.
So another question we've been getting are around family members of people with ALS or gene carriers, and are they able to participate in this study? Dr. Oskarsson, do you want to start there?
So I think that our current protocol, again, if you know your gene status, we will be directing you to prevent ALS. Whereas if you have ALS, we're able to welcome you, but and family members who don't know their gene status are welcome.
Laura, anything to add on that?
Yeah, so the criteria that is for both our healthy controls and our ALS participants. If you're an ALS participant, you just need to have a diagnosis of ALS, and be 18 or older and meet a little bit of another set of criteria. But for controls, exactly what Dr. Oskarsson said, 18 or older, if you don't have a known mutation or neurological disease at the time of consent, we are more than welcome to have you on the study. If you do have a mutation and you're asymptomatic, there are other studies. And all of these studies do help. We get strength through numbers. So the more people we have enrolling into research, the more quickly we can fight ALS a little bit harder.
Thanks, Laura. And you did mention at the time of consent. So with that, meaning over time throughout the study, if somebody finds out, they're still able to participate?
So once they've started on the study, and then let's say, they decide to get genetic testing halfway through and we find out that they have it, have a mutation. We kind of handle that on a case-by-case basis with the site. If there are the opportunities to enroll into another study that's better suited for them, then we can transition them off. But still, again, all the samples we collect, all the data we get is still valuable, and we still share it out with the research community.
Thank you. And we also have a couple questions coming in about other ways to participate in research, some examples of brain donation here. So are there other ways through Target ALS that people can get involved? Laura, do you want to get going on this one?
Sure. So our longest standing core resource is our brain and spinal cord donation. And it's been going on pretty much since Target ALS started. And a lot of the sites that are on our biofluids or natural history study are also sites that are taking those donations. And it's a great way to leave a legacy if you want to do that. You can learn more about it through our AGRI webpage or our website. And also, from a scientific standpoint, if you are part of the biofluid study or natural history study, and then you move on to donate your brain or spinal cord, that's an invaluable resource for the scientific community to see the longevity of the disease progression and even after. So that is an option that we have.
Dr. Oskarsson, could you talk a little bit more about what Laura just mentioned about postmortem tissue and biofluids and all this data and how it comes together to paint a clearer picture for researchers and how that is supporting them to identify new risk factors?
So ALS is a disorder of the nerve cells in the brain and spinal cord. We can look at it sort of by looking at the spinal fluid, but looking in the brain and spinal cord, actually at the neurons, is by far the most telling way to kind of try to understand what's going on there. And so it is even more important as a measure than the spinal fluid. Particularly for samples, if you can have somebody who has given of themselves during life, and then we get that final donation, keeping those two pieces together and looking for knowing what things actually look like in the nerve cells, and then having the preceding spinal fluid samples can be really educational, and there's a lot of insights that can be gained from that. So that is a very powerful donation.
Thank you. And yeah, of course, we at Target ALS, I know everyone on our team is so honored and know what a big gift it is for anyone to participate, even with our one-time blood draws. We take any sample given with the utmost respect and honor that it deserves. And as Laura said earlier, making sure that that sample gets immediately sequenced, the data gets out, the samples are treated as best as we can, because we are very thankful for everything that these participants are offering us here. And with that, Laura, could you talk a little bit about how scientists can request those biofluids or get access to that data?
Sure. So we have a biofluids request process, and it's on our website. You can go to the resources that we have available for scientists. And with that, you can go in, you complete a form asking, specifying what types of biofluids you would like, the rationale as to why you need them. And then it's vetted through a couple of our IRC members and conversations with our Senior Director of Scientific Programs, Amy Easton. And once it's approved, we'll establish a contract, put that in place to transfer the materials appropriately. And then the samples will be sent from our biofluid repository directly to the researcher. And again, at no cost, no strings attached. And then the biggest thing that complements these biofluid samples is our data engine, which is a really powerful resource for the scientific community, where it has everything from sequencing files, all of the clinical data that we were able to gather during these routine clinic visits. And we have multiple sets of data from different resources, and we'll have our proteomics and NFL data that's coming out as well. So there's a plethora of things for researchers to mine. And we have an amazing data scientist as well, who's more than happy to help guide you through how to use our data engine if you do request samples or want to mine anything.
Thank you. We're getting a lot of people asking about what clinics we have and where they can go, which is really exciting to see. So I will make sure to include a list of our current sites in the follow-up email after this webinar, so you guys can learn more there. But we're also getting some international interest. So could we go back a little bit and talk more about the diversity and why it's so important to have a diverse study, both racially, ethnically, geographically? Dr. Oskarsson, could you take that question?
So again, we touched on that genetics is something that where we're seeing patterns, again, leading into ALS. And genetic diversity in different parts of the world are vastly different. So to understand why ALS occurs in Scandinavia doesn't necessarily inform us that much about why ALS occurs in Asia. So again, trying to better understand why ALS occurs in general. And anytime we stumble upon one of these genes or causes, we're hoping that it's not just going to unlock that particular gene change, but ALS in general. Again, that whole sporadic piece that we're still struggling more with than we are with some of these genes. And if the more perspectives we get, the more shots we have of kind of getting to common denominators.
That's great. And Laura, anything to add there and anything to add about how Target ALS has started to expand internationally?
Yeah, so I mean, we have a couple of active sites internationally. We have our site in Bogota, Colombia with Dr. Pena, and already we're seeing exactly what Dr. Oskarsson was saying, different mutations that are not common in North America, but very common in South America. The rate of progression is different, and it just kind of echoes from a scientific perspective why we need to keep going. And so with that, we've been expanding to Asia, and we're hoping to have 2 sites in India, the Philippines, Malaysia, and all of that is to give us that better global perspective and picture of what ALS looks like in different parts of the world.
And I would add, and also the exposure side. I mean, exposures are probably also vastly different again. So we have more traction on genes right now, but we're looking at the exposures too.
Yeah, and that's a great point because we're actually adapting parts of the environmental questionnaire. For example, in India, where pollution is a lot more prevalent than maybe in some parts of the US, kind of being more targeted and focused on is that potentially playing a role with disease onset or the prevalence of ALS?
It's really interesting. And I think bringing it back to what we talked about earlier with there might be more genes that are risk factors, but just having that one gene might not cause ALS to manifest, but maybe that plus some of these environmental factors. So it seems like AGRI feels like a bit of putting in the pieces of the jigsaw puzzle together to really get that full picture of ALS. Am I speaking correctly?
Yeah, I would completely agree. And I think that's just it's why Agri exists. It's why we're being comprehensive, and it's why we're emphasizing diversity.
All right, we're coming up on time, so I just wanted to offer both Laura and Dr. Oskarsson an opportunity for any closing thoughts and to thank everyone for joining us and for staying on for the entire time. So Laura, I'll pass it to you.
Sure. I want to say thank you again for everyone that did join today and for all the questions. We have all of our sites listed on our Agri webpage. If you have any questions, you could always reach out to us at Target ALS. We're pretty responsive and filter it in real time. So I'm happy to take any more questions and everything that the ALS community does. And if you choose to be a part of the study, the impact's immediate. And I think that's the key to moving forward more quickly.
Dr. Oskarsson.
Yeah, I would also thank everybody who sat in and listened to this, and big thanks to anybody who has given of themselves to this project. I think we are saving some people now. There are people who live, but we need to get to where everyone lives.
Thank you. And thank you, everyone. We will definitely go through the questions that we didn't get through. So please keep an eye out for that follow-up email with the recording, as well as the link to those answers. And as Laura said, if you have any specific questions or need to get in touch, you can learn more on our website, send us an email at hello@targetals.org. And we will be happy to get back in touch with you and help you make decisions throughout your research journey here. So thank you all, and I hope you have a wonderful rest of your day.